Welcome back, everyone. So this is the Ask the Experts session. This is one of my favorite sessions in a conference, and we have all these amazing experts up here. I'm going to be reading the questions, and then Dr. Straub is going to select an expert that he thinks best fits the question. And so at the end, if you didn't get your question in, we, of course, want you to be able to verbally ask it here, and then we'll try to field it. So let's just get started. Okay, so our first question comes from a patient asking about AAV antibodies. This is a good question, actually. What is the implication for families with more than one affected sibling? It's a common question. Is it okay for siblings where one enrolls in gene therapy trial to have the other sibling living in the same household?
And there are more experts sitting among the audience here, and I will pick one of them as well if I feel that these incompetent people here. Anyway, so Tanya, are you?
The problem with the AAV antibodies, we have 50% of your patients, adult ones, that are positive for the AAV antibodies. So that was the main problem for enrolling patients in the gene therapy. And that will be also a problem for other gene therapies. So clearly, researchers have to think about another way to bring the gene or modify something around the gene to improve the disease or to stop the disease or something like that. Perhaps we have to think around the disease, not from the gene, but around what is around the modifiers, I don't know, something like that, because the problem is this AAV. Although they are also talking about immunosuppression, like we did in Pompe disease, starting with high immunosuppression. But, it's...
But the question is a slightly different question. I think, if I understood the question correctly, the question is, if someone is getting gene therapy, a sibling with AAV, is there a risk that a sibling in the household will develop immunity because of the adjacency to someone who was injected?
So in other diseases like spinal muscular atrophy, the AAV is shed in things like urine, feces, saliva, and tears for, depending on the study, 1 to 2 months. So we generally recommend that a sibling, not parents, siblings, avoid contact with bodily fluids for 1 to 2 months after gene therapy to decrease the risk of seroconversion. So if you're AAV negative, to decrease the risk that you will become antibody positive. So that you ultimately would be able to treat the second sibling. Or, gene therapy is becoming available for other diseases. So maybe somebody in the household would develop something else that might need gene therapy. So avoiding contact with secretions and bodily fluids for 1 to 2 months, wear gloves, minimize contact, that kind of thing.
Thank you very much, Kathy. Because after COVID, we are all virologists. It's a bit similar to this. I mean, just because you're sitting in a bus with someone at the end of the bus and he coughs, you don't necessarily, get COVID. But if you go close enough and, it's in your face, and it's a bit similar that you don't need to isolate the other sibling in, sterile conditions or so, but be, pragmatic about this, exactly as Kathy said.
That's very good answers. All right, this is a good question. Can we apply voluntarily to participate in the ribitol study? Good question.
So, Nick is the ribitol expert.
Yeah, so I think you can go to, and for really any of the trials that were mentioned, you should go to clinicaltrials.gov. They'll list the sites and the site contact, and you can just find the one that's closest to you. And absolutely, I'm sure they're looking for people to enroll in that study.
Mm-hmm. Thank you. If someone is in the ribitol study, would they be also eligible for gene therapy later on?
So you have to, this is true for any clinical trial that you participate in. You have to look at both studies and see what would be exclusionary. for the different studies. So I'm less familiar with the Atomio study, but I know, for example, for the AskBio study, that being on ribitol is not exclusionary. However, I would imagine that it would, because it would interfere with the efficacy of the ribitol study, so you probably wouldn't be able to be in both studies at once. In fact, I'm sure you wouldn't be able to.
Okay, thank you. If someone is in the ribitol study, I've already answered, sorry. Where's my brain? I'm sorry. After the gene therapy, can one have a baby? And then here's a follow-up. Will the baby have any kind of inheritance with LGMD after the gene? I think the main question is, after the gene therapy, can someone have a baby? Because they have the virus that has been running through and altered gene therapy stuff.
I'm not quite sure if I understood, but if it's, you talk about having a baby and to carry the genes forward,
it would be-
you won't change your DNA through the gene therapy.
That's what they're worried
studies.
about.
Yeah. So if you might pass on the FKRP gene defect to next generation, yes.
Okay. I think
Maybe
they were...
just
yeah, go
one.
ahead.
So it's also important to understand that for a clinical trial, in the clinical trial setting, you may well be asked to not get pregnant while you're in the clinical trial. Okay, that is important, but that shouldn't be confused as if gene therapy is approved, that if you get it, you can't have a child. Okay, but often for safety and toxicology reasons, during clinical trials, there are certain requirements that you should not get pregnant while you're getting an interventional drug that hasn't been approved yet.
Okay. This question, is gene therapy a final solution? For example, will I be able to stop going to the doctors?
Any volunteers?
We don't have enough data because there is no gene therapy still approved for this disease. But if we extrapolate from other diseases, what we expect is a change of phenotype of the patient. Not a complete cure, but sometimes it's a complete cure. But what we are seeing with others, namely with spinal muscular atrophy, is a change of phenotype. So this means that we also need to know more about what is going to happen after the gene therapy to be able to tell you, well, this problem is going to happen. So you are not going to get rid of us, and we are not going to be out of a job, I think.
Yeah, I think the other thing, especially in muscle diseases like LGMD2I, the gene therapy does not what they call integrate into the genome. So if you think about it like a computer, you're running the program from a flash drive that's in the computer. And so I think there's still a lot of questions about the long-term durability of it, or how long it'll be effective. And then the last point about it would be to say that even if the gene therapy helps to correct the genetic defect, there's still work that needs to be done to build muscle if it's at all possible. So I think think about regenerative medicine approaches as well.
So the short answer to the question was no. Kristen.
I just wanted to add something about pregnancy and being in studies, because if you get pregnant, all of us who have been pregnant know that we get bigger. And then the tests, which are going to test the effect of the medicine, will be affected. So just so that you understand that it's not because we are that afraid that things might be toxic, but it might also be the fact that it will change your body. So, yeah.
Okay, our next question is, can problems of urine retention be connected to limb girdle, specifically with females?
So we know that there is an increased incidence of urinary symptoms in people with LGMD2I compared to healthy household controls. Many of those symptoms will be modifiable by magic drugs that our friends in urology, not neurology, manage. So if you have symptoms, it is definitely worth talking about seeing a urologist for symptom management.
Yeah, very, very important advice. Yeah.
Here's a good question. Is it possible to reduce the amount of accumulated connective tissue in limb girdle?
Yeah, and this is a question that applies to all muscular dystrophies, basically, when our muscle tissue is replaced by connective tissue. Any volunteers?
I mean, I'll start by saying not today. No, I think I don't. I think one of the concerns, and definitely when we talk about gene therapies or these other really promising therapies, is that as the disease progresses, eventually some of the muscles do develop a lot of fat or scar tissue, and as best as we can tell right now, that's not reversible.
Can I just add? At present, there are no drugs that can affect fibrosis as far as we know. But there are trials and there are drugs that are being, so it's not a complete lack of hope, but it's not for now. But there are trials on drugs to try to reduce fibrosis.
Okay, thank you. Do you have any experience with the accuracy of smartwatches in terms of the medical data that they provide?
The physiotherapists are all gone, huh? Andreas is here.
Well, there's a lot of people write about different devices, they either be Apple Watches or Garmin's or whatever, and it seems like you should take some of the results with a grain of salt, but it looks like the heart rate variability might be the one aspect that is most accurate based on the data that is shown. But for example, sleep data, etc., that is highly variable, and I wouldn't trust it completely when it gives you, if you have REM sleep or whatever it is. But heart rate variability seems to have some correlation with if you're starting to have a flu or different things like that. Yeah.
Okay, thank you. Should ACE inhibitors be considered when no symptoms are present with the cardiac muscle? Should it be used as a protective medication? And then a follow-up as well to this question in a moment, but that was the first question.
We don't have the cardiologist with us anymore.
We use it as a preventive medication very early. And we prescribe it very, very, very early in the disease. So I think it could be protective.
I agree. We do know that genotype is a strong predictor of when you are likely to have onset of cardiomyopathy. So if you're homozygous for the common mutation, you are not likely to develop clinical symptoms or severe disease until well into adulthood, whereas if you are a compound heterozygote and have one of the more rapidly progressive forms of cardiomyopathy, that group, I think, based on experience with Duchenne, your cardiologist might or neurologist might recommend starting a prophylactic ACE inhibitor or might not. I think we have no data in this population. Certainly when somebody develops an abnormality on your echo that shows that or cardiac MRI that shows that you have early onset cardiomyopathy, then either an ACE inhibitor or what we call an ARB, so our standard therapy for early onset of cardiomyopathy, regardless of the reason. So definitely start a medicine if you have an abnormality. It's, I think, provider dependent whether you start something if you're in a high-risk group before you have an abnormality.
We should also point out that this is not about emergency. So whether you take it 6 months or a year sooner or later probably doesn't dramatically change anything. But I think, all the advice here is exactly important.
Thank you. There's another question. Can carriers of LGMD2I have symptoms such as muscle weakness or tiredness?
No, as far as we know, they don't have it. And I think everyone agrees on that. And as you know, we have a large community in Norway. And I think we would have seen it in Norway because there are a lot of carriers, because there are a lot of patients. So I don't think so.
Thank you very much. And just to emphasize, so Norway has the highest carrier frequency in the world for this disease, and so therefore Kristen is exactly the best person to answer this. If the Norwegians haven't seen it, it's a no.
Well, what is
know.
the carrier?
It's a question. Do,
Yeah,
do
sorry, go
the
ahead.
carrier have elevated CK? Not so much, but 2 or 3 fold. It's something, a question that was in my mind for the carriers.
I think that hasn't been tested. So well, maybe we should do that, Andreas. Get all the parents of all the patients and all the siblings. Yeah, but I don't think so. But yeah.
It would be interesting to check, and the question is really relevant, of course, for the companies as well that develop gene therapy, because if the carriers definitely do not have any symptoms, and they know, okay, if we would have at least half of the full dosage, then you don't get any symptoms.
I'm fascinated by that whole topic just now. That's great. All right. Okay. Boy, don't you sometimes just want to study something that's like, ugh, it just sounds so exciting to study something like that. Okay, our son is 16 years old. He can walk. However, he cannot get up alone, or outside he has to use his wheelchair. His heart is affected. We are interested in the D-ribose trial. How can we, living in Germany, get in contact for international trials like ribitol? What can we do on our own to get to know and get in touch with others in clinical trials?
I think, so if you're here, the folks from ML Biosolutions are here someplace. I don't see them in the room. Oh, there we go. Okay, so if you're in the room now, that person who's just waving her hand is the person that you want to ask that question to for real. And I guess you want to comment specifically about German sites or enrollment.
Get a microphone.
Yeah.
Yeah, so feel free to take my card. I'm happy to answer your question directly, but also check clinicaltrials.gov. Site information will be there, and then you can contact the site directly. And for Germany, we don't have the timing yet, but we are working potentially on getting a site there.
Okay, that's clinicaltrials.gov, G-O-V. All the clinical trials are registered there. And on this website, which is also used by healthcare professionals and so on, you can see which are the sites that are participating in this trial. And so this gives you a first idea who the best contact might be. And Nick Johnson mentioned this before as well. So that's we often use clinical trials, one word, .gov, to just look at who's actually involved from our colleagues. Why am I not involved? Okay.
Do you really? Or y'all just joking?
Just that there was one additional comment.
Okay.
You said the child was 16, right? And based on the inclusion criteria, that would not be eligible to enroll in the European sites. It would be US, UK, or Australia, right?
That's correct. Yep, that's correct.
So here's a follow-up question. If someone lives in that country, can they travel to the UK and enroll in the ribitol trial there?
So I'll answer that one because I'm in the UK and we are participating in this site. Now, in the UK, we have a lot of patients as well. And there is a discussion ongoing, I think, in many countries about fairness, why should only the local patients be the ones who are enrolled? Should we not, have a level playing field that everyone can participate? Now, but there are insurance complications, there are hospital rules as well, there are regulator rules. It's often not the investigator, the doctor, who says yes or no. If my hospital says, no way, I'll give you an example. You're in a clinical trial in a different country and you break your leg, who pays for this? The hospital says, that's not our responsibility, this patient comes from somewhere else. So it's not that easy. For some very rare trials, very rare diseases, even rarer than LGMD2I, often it is agreed already by ethics and in the protocol that you need to recruit internationally, and then it's okay. So the best thing, again, is probably to get in touch with the local site and just ask, are you open for international patients or not? For Newcastle, currently, we are not.
Okay, thank you. Here's a very good question. What is the relationship between ribitol and D-ribose?
You should answer some of these questions. No, so D-ribose is ribitol is basically a prodrug, or it's converted to D-ribose in the cells, and the reason why that was done in that way is because ribose and glucose compete for the same transporters. So taking high levels of ribose in some individuals can actually make them have low blood sugar, and that's not true with ribitol because it's a prodrug, and so it's converted to ribose inside the cells.
And keep in mind, for the study, you need to take a lot of ribose, or ribitol, 12 grams. Therefore, it for safety reasons and tolerability reasons, it's better to use ribitol.
Okay, the same person also asked, would you recommend a patient take D-ribose if they cannot get into the ribitol trial? And at what dose, they asked.
For that same exact reason. Some people definitely can have hypoglycemia, so you really need to be very careful if you're going to try and do any supplementation off-label and without your physician's advice. I think you have to be very careful with that.
Maybe just to add that it's not a secrecy that you can get ribose, D-ribose, for fairly little money compared to expensive drugs from providers online. But I think the point Nick made there is really important. If you would do this, and I'm not encouraging anyone necessarily to do it, but if you would do this, really make sure that you are under the supervision of a physician. Don't just do it alone. I'm taking this and I'm not going to tell anyone. Make sure that this is in some way monitored. Again, not that I'm encouraging anyone.
Make one more general comment as well, which is that, the reason why these clinical trials are designed in such a way is because, work has been done actually by usually lots and lots of people over many years to bring us to the point where we have therapies that we think might be effective. But we don't know that they're effective yet, and that's the reason why it's so important where possible to participate at, if you're interested in ribitol, to participate in the clinical trial so that we can all as a community come to an answer about whether or not that drug works or not. And so before you go out and take a whole bunch of ribose, it's best to kind of, allow for the trial results to really tell us whether or not that's going to work.
Thank you. This is a really interesting question. Do you have to inform your insurance company if you participate in a clinical trial?
I am aware of no obligation to inform your insurance company. They are not involved.
Usually your identity is kept secret. In most trials, your identity is actually really kept secure and secret. So, no, I don't think so.
It's also that every trial has an insurance. Okay, if anything happens in a trial, it's not your insurance company who needs to compensate. It will always be part of the trial. And in the UK, we don't have insurance companies in the same way. So it's a national health system. And you don't need to inform the national health system in the UK that you are participating in a trial.
Did somebody, was
Yeah.
there anyone else that had any?
No, I was saying that it's the same. Your identity is kept. You are confidential, and you don't need to inform. At least in France, to inform health insurance companies. They don't work the same way as in the States, so it's completely different. But the hospitals sometimes they get to know, because if there is some sort of complication and we need to do some extra tests in a hospital context, they may know, but you don't need to inform them. But the hospital is informed of the running of the clinical trial.
Maybe just one last comment. I mean, if you participate in, for those of you who do or have participated in clinical trials, this is all information that will be provided with a clinical trial, so-called patient information leaflets or forms, and they will also address who is going to be informed about this, who gets my data, who gets, personally identifiable data. So all of this is highly regulated.
Thank you. This is an advocacy question that involves the ages with the FDA and the EMA in regards to the Ribitol trial. And the question is about the FDA approves a clinical study for adults 18 plus and for children from 12 years. And I just want to make sure that that's the right. The EMA only approves the clinical study for adults 18 plus. And so the patient wanted to know, is there any options to enable the EMA to open the clinical study to 12-year-old children and up? Is there any way to advocate for that? And I think that's a good question. Is there anything options they have?
Well, we would almost like to ask colleagues from ML Bio again if there have been discussions with regulators about this. Maybe as a general comment, okay, for regulatory authorities who look into new clinical products, safety is the most important aspect. And of course, in terms of legal requirements, an 18-year-old can consent to say, I'm happy. A 12-year-old needs consent from the parents. And regulators sometimes say, look, because we don't know how safe this is, we do not want the parents to make a decision if we can enroll adults who can consent. So sometimes these are just really, formalities about who gives the consent and how much do we know about safety. But the colleague from.
Hi everyone, I'm Mallory. I work at ML Bio Solutions, and thanks for the good question. So we did try to push back on this. So EMA asked us not to go to 12, similar to what we're doing in other countries. So we did try to push back, and unfortunately for this trial, it was not successful. But we are actively in progress designing trials to go into pediatric patients in Europe and elsewhere. So that is something we are actively working on because we understand it's important. We'd like to be able to go even younger as well. So, but thank you for the question.
Thank you, Valerie.
And the other thing is that if in Europe it's approved by EMA and let's say the result of the trial is very positive, you cannot immediately transfer it to the pediatrics. You need to have a pediatric trial running, or at least in parallel. You cannot just use it because the authorization that will be given, it will be for adults over 18. So it's good to start thinking about these things now to anticipate a good result from the current trial.
Okay. With all the data collected in natural history studies, can this replace the need for a placebo-controlled group in future clinical trials? Good question.
Excellent question.
Yeah, great question. Not right now. I think that's the short answer. natural history data and registry-based data can potentially, if you have longitudinal strong data, be approved by regulatory authorities in the future for use as a placebo group, but not right now. In some ultra-rare disorders, it has happened, but for FKRP, it wouldn't be possible right now. But yeah.
So in the US, in a different disease, natural history data was used as after the phase 3 trials, so the first placebo-controlled trial, natural history data was used using a special statistical analysis thing called propensity matching to replace a second phase 3 trial, which the FDA typically requires. So there's been at least one instance where the FDA did consider natural history data as a supportive study, but didn't replace the placebo-controlled trial.
Just to add on this will also always depend on the length of the trial. So we do placebo-controlled trials because there is something that is called a placebo effect, and it's extremely complicated, which means taking something that doesn't have the actual compound can still have a positive effect, and it can actually get better if you get an injection, and even better if you get an infusion, and even better if you get surgery, but it's not lasting. So, if you do a short trial for 6 months, the placebo effect is a lot stronger than if you do a trial over 3 years. Okay, and regulators might be happier to say we'll use natural history data for a 3-year study, because placebo effect, if we look at natural history data instead of a placebo arm, because there's really not a placebo effect over 3 years. But if it's a short trial, they will never, I think, allow this.
Okay, those are all of our written questions that have been submitted. Now we're going to open up to the floor, and if you'd like to ask a question of our expert panels, just please raise your hand and we'll get a mic over to you.
The timing, Catherine, has been amazing. 4:55 is when we were supposed to end.
Oh, are we supposed to end now?
No, we can still, of course.
Oh,
We
okay.
have flexibility. So just by a show of hand, yeah?
Hi, just as a matter of interest, do you have any idea about how many patients there might be in the world, or what's the incidence of being a carrier in general populations?
Who wants to address this? Well, I'm happy to start. So the carrier frequency differs very much depending on the country. So we have heard that in Norway, there's the highest carrier frequency in the world. The carrier frequency in Norway, for some regions, is, I know what the efficacy, what the effect it is. Do you know the carrier rate? It has been published. 1 per 90, so one out of 50, roughly. Yeah, so in the UK, it's 1 in 400.
Iowa is 1 in 333, actually.
So, it's similar. So, but these are higher carrier frequencies. In Japan, it will be, 1 in 10,000. Depending again where the Vikings were. They never made it to Japan. So, LGMD2I has not been reported in Japan, even though it's a big country. So there is no global figure, really. You need to look at different countries. But, yeah, in Western Europe, I think for many it's probably similar as to North America, between 1 in 300, 1 to 500. And then you have hotspots like in Scandinavia where it's a lot higher.
Do you have any estimate on the total number of patients?
So the total number of patients, because of that challenge, it hasn't really been calculated, how many patients there are. It was interesting to hear from Kelly and Catherine that the chat group has about 2,000 patients. Not that we know whether they have confirmed diagnosis, probably. In the registry, we have about 1,000 patients now. We don't know for everyone. I would say, this is definitely in the range of maybe up to 10,000 patients worldwide. That's my guess. I don't know whether the experts.
In France, we have 150. In France, 150.
Yeah. So we have done a big study in India now as well, looking at girdles. Again, no influence from the Vikings there. So extremely rare.
I know a guy in
Maybe it's
Bangladesh.
less, yeah.
I talked to a guy in Bangladesh.
I think 10,000 might be a high number.
Yeah. Maybe it's too high, yeah. Lindsay.
This is related to you mentioned that if either of the current treatments is approved, it will only be approved for adults, and a separate study will have to be done for children. Is the same true of non-ambulatory patients? So at the moment, all the trials are only using people who can still walk, because that has to be measured. Will there have to be a separate set of studies done for non-ambulatory people?
It's going to depend mostly on the results of the trials. If they are very, very positive, probably they will skip the non-ambulant phase. If they are not very good, they will make it, because there's money behind that, money that the payer has to pay. And if there are not good results, it's difficult.
It's been asked several times on the forums, and I wanted to be able to answer people. I think people had different answers actually from different trial centres.
So we know from other trials where you definitely need to have additional studies for non-ambulant patients, because the outcome measures, so the assessment of the patient is a very different one. If you look at walking, then you can't use this in a non-ambulant population, and so therefore it's slightly different. May I just actually pass on also the question about the prevalence to the companies, because this is a big question in terms of marketing as well, and what is your patient population? Have you made any estimates, looked into this? Who is brave enough to say something? Thank you, Valerie.
Hello again. I think our estimate is similar to what you guys have been saying. I think maybe around 7,000, so similar to the 10,000, but a little bit lower is what we've found.
Yeah, about 7,003. Yeah. So in that range. Yeah.
We've got a question right here.
Yes, ladies.
Okay. I was wondering, because you try to find different way to treat this disease, but is it possible to, in the future, if this goes well with what you're, will it be possible to use different treatments together in the future? Because, yeah.
No, that's a very good question. I mean, if several drugs are approved, can you use them together?
So there is actually a study in a mouse model of LGMD2I that shows that the ribitol and gene therapy both have some benefit, but when you put them together, they are quite additive or synergistic. So the expectation would be yes.
But I think, at least from Norway, we know that the regulatory authorities will have problems if it's both the medications will become very expensive. It will be hard to get two types of medication. So at least for us in Norway, we can only use one type for the SMA patients. We cannot add another one if we don't see an obvious effect. So.
But then if you start in one medication
Mm.
and you will find the results that the other one afterward will give better results, is it possible to change from one to another?
Yeah, that will depend upon.
So the difference between LGMD, or at least these drugs, and SMA is that gene therapy is a one-time infusion, so you can't take it back. So I think the question then would be whether or not they would continue to cover the second medicine.
Yeah.
The challenge, just to also, sorry, to add on to this. So the challenge that payers, insurance companies, health systems have, okay, so you have two drugs that are tested in parallel, but one finishes before the other and becomes part of standards of care. Do you now need to show that a second drug adds any benefit? That is difficult, and the landscape will become, I hope, hopefully, a lot more difficult. We are now starting to see this in other diseases where there are certain drugs that have become part of standards of care, and so you cannot, in a clinical trial, say, we take this drug away because it's part of standards of care. But then you need to show that your drug, in addition to what the patient already gets, is still effective. And that can be very challenging.
I have a question concerning how the different research projects on LGBT, there are so many subtypes, and it's a very heterogeneous field, I understand, very complex. And we hear about different research projects on the different subtypes. So my question is there no common field? Is there no, what do you call, I understand you have some cooperation, but when you sum up this research, is there no win-win cooperation? Is so you have to look at the different subtypes for themselves? Is there, because if you have 30, 40 different subtypes, there are different processes, different reasons, causes, different, you have different genes, different proteins, etc., etc. It's very, very complex, I understand. But how is this research on different subtypes in this heterogeneous field, how is it coordinated? Is it a fragmented research, researchers doing something there and something there, or what kind of coordination? How is it done globally?
Excellent question. This is a topic for a whole conference. So for the next one, or for the limb-girdle conference. I don't know whether anyone wants to say something.
I mean, I think your question was about limb girdles overall, so not just LGMD2I. So I will tell you that the consortium that many of the people here are a part of internationally does do the same basic protocol across multiple different subtypes to begin to help us, develop a more uniform approach. And then the other thing I would say actually is that, even though we talk about these natural history studies as being all the same, sometimes you ask a question that's more specific about motor function, or maybe one really focuses on cardiac function. So, even though you do multiple different studies, they can all answer slightly different questions. And I think you're looking at a very collaborative group of individuals.
Can I add something? Is that even if this looks like isolated and isolated initiatives around one disease, we all learn from the mistakes of the past. And we definitely know that there were mistakes in developing treatments or developing clinical trials or getting some endpoints. And these results are available for the community, and we usually go back to those results and say, well, they did this here, but they shouldn't have done that, and we learn with these mistakes. That's a way of coordinating things.
Yeah, just to, if we just look at LGMD2I now, ribitol, of course, can't be used for any of the limb-girdle muscular dystrophies where ribitol isn't the problem. It's actually specifically, highly specific for FKRP. Only FKRP, which is an enzyme, uses ribitol or ribose as a substrate. So you cannot translate this. Gene therapy, the principle is, of course, something that would apply to all limb-girdle muscular dystrophies, really, the recessive ones at least. And there you can learn, as Teresina said. But let's take heart involvement, doesn't play a role in the most common forms of limb-girdle muscular dystrophy. But then there was a question about connective tissue, for example, earlier, which we call fibrosis, which is something that all the limb-girdle muscular dystrophy shares at a certain stage. And you could say, well, maybe there's an antifibrotic drug that could be applied to many limb-girdle muscular dystrophies. Heather.
Just to add that this is actually the second meeting that I've had the pleasure of organising in Amsterdam, and the first one was precisely to try and remove fragmentation. So we had a group of us meeting in 2019, looking at the different outcome measures to ensure that there was some compatibility of the various natural history studies that were ongoing.
And Volker, we have one question here.
There's a microphone as well.
Yeah, so I have a question in regards to insurances. So do you see any risks for the future that the health insurances are not willing to pay the costs for the therapies anymore? Because we can see this in the countries, for example, Germany, the monetary budgets for health insurance are getting tighter and tighter, and there are a lot of negotiations with the health insurers. So do you see a high risk? And if yes, what can we do, maybe as patient organizations, to work for this, to make this continuously happen?
I'll pass this on to the experts as well, but just to be clear, in the drug approval process, there are two big steps. One is that the regulators that look at the safety and efficacy of a drug need to approve a drug. They don't think about the price, and they may say, this drug looks safe and effective, and we approve it. And then the company says, well, it costs 2 million. Now, the second step of approval means the people who actually have to pay the 2 million, the payers, we have mentioned the payers. So these are, depending on the health system, either insurance companies or a national insurance as well. They then also go through this process and say, okay, it's safe and it's effective, but actually it's not worth 2 million. And that's your question. So is this going to be a bigger problem?
I'm only answering on a European point of view. One of the things that we saw that was a success in Europe was with the COVID vaccination, where the negotiation, even if the European Commission didn't have the expertise at the time, for the price of the vaccine was done collectively in Europe. And this made the price come down. What we see now with all these therapies that are coming, gene therapy, and not only gene therapy, is that the price in Europe is negotiated in each country. And I can tell you that there are lots of lobbying at the level of the European Commission, lobbying from the patients organizations in general, like Eurordis and others, to make the negotiations central as they were for the COVID vaccination. And this is the best way to try to have uniform prices and lower prices, at least in Europe.
In the EU, because the UK is still Europe, in the European Union. Yeah. Yes. Yeah.
But Teresina, it's not the case, actually, because for the European Union for this drug, not the case, because only paracetamol is not also this simple drug. There is different prices among the countries, so there is a big problem with all of these drugs that we use every day. But it's just thinking about SMA. It was open for all diseases in France, 1, 2, 3, even if we are very disabled or not disabled. But, however, the social insurance says, yes, we open for every kind of disability, every kind of people, but we will show if there is an effect for disabled people, very severe disabled people in this drug. Within 3-4 years, you have to provide the data, and we will see if we reimburse still the drug for those people. So, it's open, but they also need to have some points of results and data to see if they continue or not.
Yeah, it's a very complex topic, as you can tell. And clearly, all health systems across the world are facing the challenge that more and more rare diseases are currently developing drugs or in rare disease areas, and that the payers will struggle. At the same time, we have changes in the demographic landscape, dementia, and These are the real health challenges, so it will be difficult. There's no question about this.
I just want to say that we all have to work politically, and we have to show that it's not only cancer that is disabling, that's not only MS in the neurological field that is disabling. We have to show that these disorders actually is present, so that the government and our colleagues know about it. So go to your local patient organization and get active. That's my advice.
And the companies will probably try their best also to collect data. Well, how expensive is it for a health system to look after a patient with limb-girdle muscular dystrophy R9 without any drugs? That's right. Okay. And if you think about, well, I have to give up a job as a mom because I have to look after the child, it's, you know, completely there. Yeah, so, yeah, but I mean, this is about health and social care. And so if you really try to figure out how much does it cost to have LGMD2I, and then you can at least say, okay, our drug needs to, of course, be more expensive than that because we also need to produce it in everything. So this is a whole field of so-called health economics. This is something that is challenging in rare diseases as well, but there's a lot of effort now going into really understanding this. And maybe just one last comment, because when you hear about these expensive, those super expensive drugs like SMA, you know, the list price in the UK is currently 1.75 million for Zolgensma. And the payers assess actually on very sober figures how much worse is a life from birth to, you know, the end of life. And there are good numbers about this, and, you know, insurance companies have these numbers as well. This is what health insurance companies figures are based on. And then if you say, okay, we invest very early in a neonate and give gene therapy, and this will become a taxpayer and will pay taxes, you know, the drug price can be higher. I mentioned this yesterday. If once you're 60 or 70 and you get gene therapy, you know, the payers might look at this as slightly different because, you know, how much do you still actually contribute to the system? But that's important. I mean, you know, this is actually how payers have to think. So it's complex.
Okay, I think we'll take one more question and then we're going to end for Ask the Expert.
Do you think that robotica, for instance, step robots, will be able to help us in the future?
So exoskeletons, and I'll ask the physiotherapist.
There's a lot of interesting advances in the field of exoskeletons. Right now they're big, bulky, heavy, hard to get in and out of. But I think in the future, if we have the same technological advances that we have, that it might be a possibility for people with walking disabilities, that they might be able to have a better function through some sort of exoskeleton. And we've seen some wheelchairs here with like primitive exoskeletons where you have systems raising your arm, for example, helping you even though you don't have any muscle strength. They're still complicated and not too smooth, but things happen really rapidly. And this is research that's, there's a lot into spinal cord injuries, for example, that leads the way, and some of the systems there will probably also be usable in the NMD field. So yeah, I think so. But at what time? I don't know.
Okay, guys, thank you so much. Will you guys give a round of applause to our experts for taking their time?
So
if you do get a chance, you know, write a sweet note and thank our experts. I'm sure that, you know, just words of appreciation on a letter or something would be so meaningful. And just think about, you know, how grateful we are to have them. I mean, our lives are so enriched because of these clinicians and this wonderful conference that you have put on. Bravo, I think it was excellent. And tonight we're going to have dinner. Will you please let us know where the dinner location is? Okay, it's right in here. So we will see everyone back at 7 p.m. And you guys go and have a nice rest, and we'll see you then.
Just before you leave, because, I mean, according to your agenda, I was supposed to make closing remarks. Hey, I mean, that's fine.
I am so sorry, Volker.
No, I'm
Make
just kidding.
closing remarks. I'm so sorry.
I'm just kidding, actually. I mean, you know, I could have just stopped here, but, well, you have already started thanking the experts as well. But I should also point out, you know, so this was supposed to be a slight summary brainstorming. One of the most important reasons why industry should invest into a rare disease is a strong patient voice, definitely. And in LGMD2I, there is a very strong patient voice. And it's fantastic to see that there are patient advocacy groups from different countries actually joining us here in Europe, and that colleagues from the US are supporting this as well and join forces here. It's extremely important when the expert answered some of the questions, you know, how do you talk to regulators, to payers? They will listen more to patients than to us experts sometimes. It sounds strange, but this is exactly how it is. You know, it's really important that the patient voice is heard. And I think it was fantastic to see. I participated in the session of the young adults and teenagers to see that the networking and the patient voice was already heard very loudly from early on, which is, again, really encouraging, and I can only applaud you to initiating this as well and continue this. I think LGMD2I, among the long list of 33 limb-girdle muscular dystrophies, is one of the best prepared for what's coming. We know that for the affected patients, the clock is ticking in a different way, and if we say, well, maybe wait for the results of the phase 3 trial, which only takes about 3 years, you think, oh my God, I mean, this is far too long. Nevertheless, I think what we have learned as well, that it's not just about the drug treatment, that it's also about ventilation, that it's also about physiotherapy, cardiac protection. So there's a lot that everyone can do, even if you don't, if you're not involved in any of the interventional trials. And I think it was also mentioned here, it's very important to keep in mind that's human research. These interventional trials aren't treatment studies, they are human research. And they may not work, or they may work but will not get approved by the payers, for example. So most importantly, and this is why the experts met already yesterday, is that we want to provide care guidelines as something that everyone can do. And so we'll work on this. We'll make sure that we involve the advocacy groups, hear the patient voice in those care guidelines as well. And then the last thing probably to say is, so what's next for the European LGMD2I community, and do we meet again? I think this was definitely a success. We will probably and hopefully get feedback about this as well, if people think, well, let's do it again, or we say, well, we have to wait, well, let's wait another 5 years and when the trials are finished. But so I think it was really great to see that there was participation from everyone. Thanks again to Heather and also to Lindsay. For me personally, actually, seeing Nina Hoffmann yesterday was one of the highlights. We first met 25 years ago, and when she yesterday said, Volker, Mensch. And I said, Nina Hoffmann, I can't believe it. So it's these personal relations that you form that are lasting as well. that really contribute to quality of life, and that's what ultimately I think counts. So yeah, I think we'll have a wonderful evening today as well later on now, and looking forward to seeing people again. Thank you very much.